Hello everyone. A very good evening to all of you. So I'm again here with another session from Unit 2 under Brahmastra Series, that is Unit 2A and 2B. I hope so I'm audible to all of you and my screen is also visible to everyone, right? Yes, good evening Anusha, good evening Sunshine, Kirtana. Okay, thank you for confirming. So, in our last session, if you remember, we had discussed with membrane structure and function. Today, we will be focusing on solute transport as well as cell organelles and protein targeting. But I have two questions from membrane structure and function, which is a very rare question and it was not asked for the past many years. Okay. So, today, the first question will be from that portion. Both sphingomyelin and phosphoglycerides are phospholipids. Which one of the following statement is not correct? One has a fatty acid tail attached via an ester bond, while the other is attached via an amide bond. The hydrophilicity of both is dependent on the phosphate group and the other head groups attached to phosphate group. Only one of them will contain CC bond and both may have choline as the head group. So according to you, among these four statements, which one is incorrect? Now here you have to remember the structure of sphingomyelin. It comes under which category of lipids? Phosphoglycerides. What it is there? What is the structure? Try it out if you are able to answer this question or not. This was asked not recent past but some years back. And this was a unique question because this was the question related to structural difference among the lipids. Is anyone able to answer this question? Yes, okay, I can see MRD Revolution has given the answer. They are asking, remember one more thing, they are asking the statements which is incorrect. Okay, so one among them is incorrect. Okay. Okay, Kirtana, Anusha, Sunshine, Surender. Any other answers from your side? Yes, Madhumita. Okay, let me give you the answer. The correct answer over here is only one of them may contain a CC double bond. This is the incorrect statement among the four because there are chances both sphingomyelin and phosphoglycerides can have CC bond. Now, can anyone tell me sphingomyelin belongs to which category of lipid classification? Sphingomyelin belongs to which category of lipid classification? Anyone remembers? And what about phosphoglycerides? If you remember this classification, it will be easy for you to get the answer. So I am asking. No? No one knows? Okay. Then another question I am asking over here. Here, in the first sentence, it is mentioned that the fatty acid tail is attached via ester bond in one of them, and another one is attached via amide bond. Can anyone tell me which one is having the ester bond and which one is having the amide bond among sphingomyelin and phosphoglycerides? Yeah, this is an example of a sphingolipid. Which sphingolipid? Phosphosphingolipid or glycosphingolipid. You are saying glycosphingolipid. Okay. Okay, let me give you the answer. See, we have sphingomyelin structure over here and we have phosphatidylcholine over here. Now, phosphatidylcholine becomes an example of what? Glycerophospholipids, or we can say phosphoglycerides, which is one of the most abundant lipids present in the biomolecule. And if you see the sphingomyelin, it is having this amide bond. Okay, and the head group is phosphatidylcholine over here. So, this belongs to glycosphingolipid or phosphoglycolipid. Can anyone tell? This belongs to phosphosphingolipid or glycosphingolipid. You're correct, Anusha. Yes, absolutely correct, Surendra. So this belongs to which category? This is, we call it as phospho sphingolipid. Now, why we call phosphosphingolipid? Because here you are having the phosphate group attached, and this phosphate is connected to a lipid component. If it is a sphingoglycolipid, then you will have a sugar molecule attached over. Okay. So now you can see here you are having C double bond, and here also you are having C double bond. So obviously both sphingomyelin and phosphoglycerides may contain the C double bond. That's why this is the incorrect statement over here. And the fourth statement, what they were saying, both may have choline as their head group. Yes, right? If you take into consideration the phosphoglycerides in general, you are having an X group, and in that X group only you are adding phosphatidylcholine, phosphatidylethanol amine, serine, etc. So obviously this is the C double bond. Okay, so this becomes the correct answer over here. Next, now this was also a kind of numerical question, not asked recently. It was asked long time back. Lipid rafts are involved in signal transduction in cells. Rafts have a composition different from rest of the membrane. I guess you might be aware what is the difference. Rafts, when isolated, found to have a cholesterol to sphingolipid ratio as 2 is to 1. And the estimated size of the raft is known to be 35 nanometers square. Now, if we take into consideration the surface area of cholesterol as 40 angstrom and sphingolipid as 60 angstrom, you need to identify how many cholesterol and sphingolipids are present in one raft. See, basically from cell biology, numerical questions are very rare. Even if you go through the old question papers also, you will get hardly one or two questions that is numerical based has come in the exam. This is just one of them. Can anyone able to try it out if you are able to answer or not? Anyways, I'll provide you the explanation for that. Lipid raft related questions are very much frequent in the exam. Okay, remember its composition, its properties, then types of lipid rafts. These are should be considered when we are going for CSIR exam. Yes. The clue is provided over here, 2 is to 1 ratio, and the estimated size is also given. Surface area of cholesterol is mentioned, sphingolipid is also mentioned. You have to identify the number of cholesterol molecules and sphingolipids that is present in one raft. Are you able to identify? Anyone is trying out? Okay, Sunshine has tried out 22, but that is not there in the option. 50 and 25, 200 and 100, 40 and 20, 20 and 10. Okay, you were saying option 2. Okay. What about others? Is there anyone who is trying out this question? See, let me give you the answer. Okay, let me give you the answer. See, basically, these are, as I told you, these are very rare questions. Okay, just try to understand. So, the correct answer over here is 50 cholesterol and 25 sphingolipid. Let's check it out how we got 50 cholesterol and 25 sphingolipid. Initially, what we are having, the estimated size of the raft is 35 nanometer per square. But the remaining surface area of cholesterol and sphingolipid is in angstrom. So you need to convert this nanometer square into angstrom first, then you can go for calculation. Okay, Ashu? Yes, you are correct. So let's find out how does it works. We know that 1 nanometer square is equal to 100 angstrom square. Okay. Now, if you want to convert this 35 nanometer square, all you need to do is just must multiply it with 100. Right? And what you will get? 3500 angstrom square. So this you can consider as an estimated size. Estimated size of what? Raft. Now let's move on to the calculation. So you remember we had mentioned in the question 2:1 ratio. So let's consider number of cholesterol as 2x and sphingolipid as x. Okay. Already, area of cholesterol is given 40 angstrom, that is surface area. So if you consider total area of the cholesterol, just 40 into. So what you are going to get? 80x. Similarly, area of sphingolipid is already given in the question 60 angstrom. So in the 2:1 ratio, we have taken this as x. Simply multiply. What you get? 60x. Okay. Now, if you take into consideration the total area like this, so what you will get? 80x plus 60x, 140x you are going to get. And we are already known fact that it is the like total area of raft is 3500 angstroms. So simply put over here. So once you get x is equal to 25, all you need to do is multiply it with 2. So you are getting 50 and 20. Are you able to get me, everyone? Are you able to understand this calculation? Yes? Able to follow? Got it? Okay, I hope so you understood where you made the mistake, right? Okay. So this is very easy question. Usually the answer will be there in the question itself. Just you need to give a twist. Okay. If you know this conversion, the things will be more easier for you. Okay. So that's it. These two questions, which I think somewhere it is a unique question from membrane structure. So I shared with you. Next question. Out of the list given below, you have to identify the lipid bilayer permeability in the increasing order. Okay. Nitrogen, ethanol, water, glucose, calcium, or RNA. Or it may start with water, glucose, ethanol, nitrogen, calcium, RNA. Calcium, RNA, nitrogen, ethanol, water, or glucose. Or the last option is ethanol, RNA, calcium, water, glucose, and nitrogen. You have to identify the increasing order, which is more permeable to the lipid bilayer. I think in the Lawdish book, you are having this permeability chart. So you can go and refer over there also if you have any confusion. Are you able to identify the lipid bilayer permeability? So among these components, which do you think is highly permeable to lipid bilayer? Is it a glucose? Is it a water molecule? Is it a biomolecule, ions, gases, etc.? Okay, I can see answers coming up. Revolution 4. Madhumita, you are saying option 2. Anusha and Ashu, Kanka, Sunshine. Okay. Kirtana. Okay. Is there any other answers? Anyone else would like to give your inputs over here? This is a very easy question and usually a direct question. Based on this, we can also expect questions in the form of statements, combination statements, where they will provide you polar molecules will diffuse, non-polar molecules seek the help of transporters, like that. So you have to identify the correct combination. Okay, so the correct answer over here, as majority of you has mentioned, answer 1. Now, why answer is 1? See, this is the permeability chart for the plasma membrane. If you see, gases are highly diffusible. So, whether it is carbon dioxide, nitrogen, oxygen, it does not require any support. It is permeable. If you take into consideration small uncharged polar molecules like ethanol, they are permeable. Urea and water, they are slightly permeable. So what is the specific channel for the movement of water? What is the specific channel that is responsible for allowing the movement of water across the membrane? I think we had discussed this question in my last session as well. Yes, absolutely correct. We call it as an aquaporin. Then followed to that, we are having large uncharged polar molecules like glucose and fructose, which is impermeable. Even ions are impermeable. Even polar molecules are also impermeable. So, how they will be transported across the membrane? They will take the help of either transporters or the carrier proteins or the channel proteins. Okay, right? So if you remember this order, you can easily fetch a good mark if a question comes from you. This is important to remember the sequence in which the molecules are permeable across the membrane. Okay. Next question. Insulin increases facilitated diffusion of glucose in the muscle cells by phosphorylation of glucose transporters, translocation of glucose transporters containing endosomes into the cell membrane, inhibition of the synthesis of mRNA for glucose transporters, or dephosphorylation of glucose transporters. What should be the correct answer over here? Anyone remembers how exactly this glucose molecules are transported in the muscle cells? Okay, I can see some answers coming up. Okay, okay, which or how the glucose molecules are imported to take up the glucose? What is the kind of transporters they are having? They have GLUT transporters, right? And specifically speaking, which GLUT transporter is responsible for transportation of glucose in muscle cells? You are having different GLUT transporters like GLUT1 is there, 2 is there, 3, 4, 5. Among them, which GLUT transporter is responsible for transporting glucose in muscle cells? See, as I can see, some miscellaneous answers came over here. The correct answer over here is translocation of glucose transporter containing endosomes into the cell membrane. Now let's check it out how this translocation takes place. Remember, it is your GLUT4, okay, into a myocyte. So when exactly the insulin is activated? When our body cells start secreting insulin. In what condition? Specifically, when we are taking a carb-rich food, right? Yeah. And when we are taking carb-rich food, what happens? As mentioned by Kanaka, yes, our blood glucose level will be increased. Yes. So when this blood glucose level increases, what happens? Insulin will be activated. It will go and bind to the corresponding receptors present within the plasma membrane. Meanwhile, whatever the GLUT transporters are present within the endosomal vesicles, slowly, gradually, they will move across the plasma membrane and get itself fused. So, they are ready to take up the excess of glucose. Contradictory to that, so this happens when the insulin is more. When the insulin level drops, that means we can say that blood glucose level has came back to normal. At that time, what happens? These GLUT transporters will slowly released from the membrane. How? By means of endocytosis. Endocytosis taking up inside. Exocytosis means taking the content outside. So here, this particular portion will be taken up inside. And the smaller vesicles will fuse with the endosomes. So when required, when they get a signal, slowly they will be pinched off from the membrane and moves towards the plasma membrane undergoing fusion, take up the glucose, and once the blood glucose level backs to normal, it will be recycled back. This is what happens, or this is what the regulatory mechanism of insulin by GLUT4 transporters within the muscle cells. Okay, I hope so. This image you are able to understand what exactly it is, right? Okay, moving on to the next question. During receptor mediated endocytosis, apolipoprotein B on the surface of LDL particle binds to the LDL receptor present in the clathrin coated pit. Now, this complex, that is the receptor LDL complex, will be internalized by endocytosis, transported or trafficked to lysosomes. And this LDL receptor is finally recycled. But a patient reported with familial hypercholesterolemia. So, what could be the reason behind it? Mutation in the LDL molecule, defect in the recycling process, mutation in the receptor, or defect in cholesterol binding to its receptor. What should be the correct answer? In fact, indirectly, what they are asking you, what is the problem with familial hypercholesterolemia? Why the person suffers from this disease? Try to change the settings, Saiba. Okay, very good. I can see all of you have given the correct answer so far. What about others? What about others? Very easy question. Again, this is a 4 marks question. The only thing is that you have to aware of familial hypercholesterolemia. Okay. The correct answer over here, as mentioned by majority of you, is mutation in LDL receptor. Can anyone tell me what is the conserved amino acid sequence present within this LDL receptor? Conserved amino acid sequence present within the LDL receptor. Because without that conserved amino acid, it will be difficult for your LDL particle to go and bind to the receptor. What is that amino acid? Yes, you are absolutely correct, Anusha. It is NPXY. Okay, this is the conserved amino acid sequence present within the receptor. What happens here? The LDL particle goes and binds to the receptor. Which receptor? LDL receptor present within the membrane. Clathrin coated pits will be constructed. So it will form an invagination like this. It will pinch off from the membrane. Coated vesicle is separated, undergoes uncoating, moves from early endosomes to late endosome. Here what happens, you are having low pH compared to early endosome. So the receptor and the ligand will be separated. Receptor will be recycled back to the membrane to take up another set of reaction, whereas your ligand will be released into the lysosomes. Why? Because lysosome is considered to be a suicidal banks, which is having hydrolytic enzymes that is responsible for breaking the oligomeric compounds into monomeric units so that it can be utilized by the cell once again. Okay. Is it clear now, Saiba? Are you able to visualize now? Okay. And one more question over here from this image only, meanwhile. How this clathrin and adapter proteins are formed? How the clathrin and adapter proteins are constructed? Anyone? Or what is the basic structure of a clathrin protein and an adapter protein? We have discussed this in the class, if you remember. The images, heavy chain, light chain, globular head. Are you able to remember? Yeah, that's the same thing I was asking Sunshine. How this pit is being constructed, or what is the structure of this coated pits? They are synthesized in the. Yes, yes, that's the point I wanted to hear from you. It is known as triskelion structure. Why do you call it as a triskelion structure? Because it is made up of three heavy chains, three light chains like this. Okay, these are the heavy chains, and the light color which you see, that is the light chain. And these are the globular heads for attachment. Attachment to whom? Attachment to this adapter protein. Now, if you take into consideration this adapter protein, it is having different subunits like mu chain, beta adaptin, alpha adaptin, sigma chain. Among this, what are the two most important components? Mu chain and beta adaptin. Why? Beta adaptin is responsible for binding to this clathrin triskelion. whereas mu chain is responsible for binding to the plasma membrane. Okay, so combining these two structures together, we are generating a lattice like this. I hope so you are able to remember, able to understand how exactly this clathrin coated pits are constructed. Yes, everyone? Okay. So, now one more question over here. How these adapter proteins are recruited to the membrane? Do you remember how the adapter proteins or who is responsible for recruiting the adapter proteins to the plasma membrane? Because if you see in this structure, the blue color one is the first to be recruited in the plasma membrane. So that means adapter proteins are first recruited, followed to that your triskelion structure is formed. So who is responsible for recruiting the adapter protein to the plasma membrane? Do we have any specific molecule, any lipid component that is responsible for recruiting this adapter protein to the membrane? Do we have? See, I have another image for you. But you can see, this is your adapter protein, which is connected to clathrin triskelion by beta subunit. And via mu chain, it is connected to the receptor over here. So this is your receptor, this is your ligand. So receptor to ligand interaction takes place. So receptor is connected to adapter via whom? Via mu chain. No, translocone is a different one, Ashu. Translocone is responsible for translocating a protein from one part of the membrane to the other. Okay, translocone will not. So here we are having phospholipid, PIP2, phosphatidyl inositol diphosphate, which is responsible for recruiting this particular adapter protein to the plasma membrane. Okay, I hope so. Now you are able to recollect, right? So if you understood this image and if you understand this image also, then can you tell me who is responsible for pinching this vesicle from the plasma membrane? See, one simple image and you can expect a number of questions from here, right? And whatever I'm asking you was already being asked in the exam previously in the form of 2 marks, 4 marks, like that. Okay. So, which protein is responsible for pinching this vesicle from the plasma membrane? Which protein? Any cytoskeletal protein? Yes. Absolutely correct. Ashu and Anusha. We are having dynamin protein that is responsible for connecting to this neck of the lattice through which it will be helping in pinch off from the membrane. So once they are pinched off from the membrane, what happens? This corresponding vesicles will be separated from the membrane and this leads to what? This leads to translocation. Means they will go and fuse with the membrane. I mean the contents will be released that will go and bind to the endosome. Endosome, because of the low pH, they will be helping in the dissociation. Okay, this is what the role of dynamine is. And this requires energy in the form of GTP hydrolysis. If in case of GTP hydrolysis, GTP gamma S is used, in that case what happens? It will not be pinched off from the membrane. So it will be encircling the stalk like this. Just keeps on polymerizing. So if it is not polymerized, the vesicles will not be removed from the membrane. That is the significance of GTP gamma S. In fact, this is considered to be non-hydrolyzable analog of GTP. Basically, these are used for experimental purposes. Okay, not used in general because this is what actually happens. Okay, I hope so. The concept behind the Clathrin pits, the role of dynamine, LDL cholesterol, everything is clear now, right? Shall I proceed ahead? Or is there any confusion in any of these images? Any connectivity link you are losing? No, right? You are able to understand? Yes? Okay, fine. So, before going to the next topic, next question, just wanted to remind you about the WISE scholarship program that is for biotech graduates, postgraduates, and professionals. Remember, the deadline is 31st July, so you have to hurry up. So, through this scholarship, one can get 100% scholarship as well. One can get 90, 80, 50, or even 25, and through this scholarship you will not have you will have access not only to the classes, that is CSIR classes or GATE classes, but also you will be having access to a number of smart tools that will help you to revise the topic easily and quickly and retain it for a longer time. About to that, you can have access to internship, workshops, and many more. Okay. So if you have any query, kindly call to us in this toll-free number, either of this number, and get itself right. Okay, fine. Moving on to our next question. Okay, so this question is related to aquaporins once again. And this was a question being asked in the recent CSIR net exam. They are integral membrane proteins in the major intrinsic protein family, absent in the bacteria. A highly conserved NPA triad is present in the N-terminal half of the protein. A highly conserved NPA triad is present in the C-terminal half of the protein. Now, according to you, which statement is incorrect over here? The recent exam, you got this question. According to you, so try to recollect the structure of aquaporins that we have discussed in our last session. Okay, I can get two answers coming up. Oh, I can see majority of you are saying as fourth as an option. They are asking you a statement which is incorrect. Vigneshwari, Aruna, Kirtana, Durga, Surender, Tanaka, Sunshine, Ashu. Anyone else? Think once again. I know there should be a confusion that NPA two times it is given. So one of them can be a correct answer. That tendency will definitely come. But if you remember the structure of aquaporins, things will be more clear. The structure of aquaporin. Do we have NPA sequence? So if we are having the NPA sequence, where exactly it is present? N terminal, C terminal, or both side? What should be the answer? And in my last session, if you remember, I have shared a table also with you. A table with respect to aquaporin channels present in different organisms and aquaglyceroporins present in different organisms, if you are able to recollect. So from there, anyone able to get any answer, any clue? Okay, let me give you the answer. The correct answer over here is they are absent in bacteria. Do you think so? They are absent in bacteria? They are present in prokaryotes. If you remember, I had shared a table with you in my last YouTube session, Brahmastra session. And there I have shared the table where E. coli was mentioned. E. coli is also having a specific aquaporin. So not only mammals, plants, yeast, E. coli, everyone is having this aquaporin channel. See, you remember A. aquaporin Z. This was the aquaporin present in E. coli. And another one was GLPF. GLPF stands for what? Glyceroaquaporins. That means that is having glycerol permeability. Okay. So primarily involved in the transport of glycerol, also facilitates water across the membrane. So we can say it is widely distributed in organisms from humans to bacteria. Now, if you look into the structure of aquaporins, basically they are tetrameric in structure with four monomers coming together creating a channel. So this is one monomer. Like that, three more monomers are there. And each monomer is having six transmembrane helices. So 1 to 6. And you are having A to E loops. This conserved NPA loop is present one towards the N-terminal site and another is towards the C-terminal site. So obviously the statements are correct. If in case the NPA is present over here, then yes, you are correct. It should not come towards the N-terminus. But since it is coming between 2 and 3, that's why you can consider it as an N-terminus. So that's the reason the statement is correct over here. A highly conserved NPA triad is present in mentioned C, N-terminal half of the protein. And here C-terminal half of the protein. Obviously it is present, right? The one NPA is present directly in this C-terminal. Another one is present in a loop that is near to the N-terminus. So we can easily opt for this answer. Okay, I hope so you got it. Next question. Some statements regarding the process of autophagy are given below. Autophagy occurs when cell contains aggregated proteins. Autophagosomes fuse with any organelle. Autophagosome is a single membrane structure. It can fuse with lysosomes forming autophagolysosome. Now you have to identify the correct combination of statements among this group. Find it out. Able to remember the autophagy events, the sequence in which different structures are created over here. Okay, you can see Ashu and Rahul has given the answer. And Sharma, Kirtana, Sunshine. Anyone else would like to give your input? Vigneshwari, yes. Read the sentences properly. Then we will definitely get the answer. So far, I am very happy to see that all of you has given the correct answer. Yes, it is D and A. So if A and D are correct answers, then why B and C are incorrect? First thing is that autophagosomes will not randomly fuse with any organelles. It can only fuse with lysosome. Autophagosome is not a single membrane structure, rather it is a double membrane structure. That is the reason. So if you see over here, this is the image. This is vesicle nucleation. Vesicle nucleation in the sense what? Vesicles are being constructed. That means that membrane is being constructed. Vesicle elongation. Finally, a complete organelle. You call it as an autophagosome, which fuses with the lysosome. Lysosome is having hydrolytic enzymes. Now, they will be responsible for docking and fusion and finally breakdown of the contents. This is what the sequential steps involved in autophagy. Now, if you look into this image, see, you got an autophagosome structure. Initially, the double membrane which is being donated, you call it as a phagophore. Okay. Now, after that only this autophagosome is constructed. Now, there is an intermediate organelle may be constructed between endosome and the autophagosome. That is known as amphisomes. How amphisomes are created? Sometimes this late endosome may be fused with autophagosome and they create a organelle known as amphisome. In fact, this is an intermediate organelle before the lysosome formation. As you know, endosomes are something that undergoes maturation, finally giving rise to lysosomes. So this early endosome, late endosome, amphysome, and finally you are getting the lysosome. Okay, so if anywhere you come across the term amphysome, don't get confused. It is just an intermediate organelle formed before the lysosome. So after that, they will undergo fusion and the contents will be released. Here MVB is mentioned, right? What is this MVB? This is nothing but multivesicular bodies, or you can also call it as multivesicular endosomes. That means these are the other names of late endosomes. Okay, remember this point. And another term which is given over here, exosomes, means if any vesicles is released out of the cell, you call it as an exosomes. Okay. So let's check out the steps that are involved over here. See, autophagy begins with the formation of phagophore. Phagophore is a double membrane structure, or we can say an isolation membrane. That is the vesicle nucleation step that we have seen in this image. Correct. After that, what happens? The concerted action of autophagy core machinery proteins at the phagophore assembly site. Phagophore assembly site is the region where the phagophores are generated. Phagophores are what? They are nothing but a double membrane structure. This leads to expansion of phagophore into an autophagosome. We call it as a vesicle elongation. Vesicle elongation. Slowly, gradually, it will elongate and become a molecule like this. So, this autophagosome can engulf bulk cytoplasm non-specifically. It can randomly take up, but it can take entire organelles or target cargo specifically. That means that is targeted to be degraded. Now, when this outer membrane of autophagosome fuses with the endosome, it becomes an amphysome. Now, amphysome is a structure that is formed before the lysosome. Otherwise, it will directly fuse with the lysosome without undergoing amphisomal intermediate. So that structure becomes autophagolysosome. So finally, what happens? The sequestered material will be degraded inside the autophagolysosome and they will be recycled back to the cells for further usage. Okay. So remember autophagy. This question was a question came in the recent exam, not in the last exam, but prior to that, I guess. 2022, I think you got a question over here. Okay, so autophagy is one of an important topic from examination point of view, specifically the sequence of events. And every time, like alternative years, they are asking this question, and like question based on autophagy. So don't skip this portion. Okay, next. Select a cellular body which is not a part of nuclear bodies. I hope so you are able to recollect what are nuclear bodies. P bodies, nucleolus, Kegel bodies, interchromatin granule clusters. Select a cellular body which is not a part of nuclear. What are nuclear bodies? What are nuclear bodies? How can you define nuclear bodies? You can see Rahul, Kirtana, and Anusha has given the answer. Absolutely correct. Nuclear bodies, we call it as a membraneless structures. That means they do not have any defined boundary. The way nucleus is surrounded by a double membrane structure, these small structures do not have defined boundaries. So, what should be the answer over here? P bodies, nucleolus, Kegel bodies, and interchromatin granules. Yes, Kirtana, yes. So, I can see everyone is giving the correct answer. Is there anyone else who want to try this question? Very easy question. See, the correct answer over here is P bodies. Now, why we are opting for P bodies? For that, you need to understand what is state terms 2, 3, and 4. What is the function of nucleolus in general? What is the function of nucleolus in general? This is the major ribosomal RNA synthesizing machinery and ribosome assembly. Right? Then interchromatin granule cluster, somewhere it is related to nucleus. So because we can predict that chromatin is given. So obviously chromatin is present inside the nucleus. Then what is a confusion is P bodies and Kegel bodies, right? See, P bodies are RNPs, that is ribonucleoproteins found in the cytoplasm, and they are composed of translationally repressed mRNAs and proteins associated with mRNA decay machinery. Okay. Interchromatin granule, as I told you, is a cluster in the nucleus of a mammalian cell, which is enriched with pre-mRNA splicing factors. Because once transcription is done, what is the next thing? Post-transcriptional modification. So that involves the splicing machinery and all. And they are located in the interchromatin regions of the cell nuclei. They appear as irregularly shaped structures that may vary in size and number. Apart from this, Kegel bodies are what? These are responsible for snRNP assembly. That is ribonucleoprotein assembly, small nucleolar ribonucleoprotein assembly. Then, apart from that, you are having clastosome, speckle, PML body, polycomb body, histone locus body. So every time You can expect a question from here, if not every time, alternatively, either in the form of 2 marks, mostly in the form of 2 marks. Sometimes you can also expect in the combination, statement based. Okay, match the following. I don't think so. They will be asking. Either they will be asking in the combination statements or in the form of 2 marks. Like they will provide you the function, ask you the bodies, or they will provide you the bodies, ask the function. Either of the way is possible. Okay, so that is the thing. Next. Now this is with respect to nuclear transport, protein targeting. Thousands of proteins are synthesized in the cytoplasm, imported into the nucleus through NPC every minute. These proteins contain NLS that direct their selective transport into the nucleus. So the nuclear import requires what? A small monomeric G protein RAN, a nuclear transport receptor that interacts with NLS on a cargo protein, gap bound to the chromatin tethered to the nuclear membrane, Jeff bound to the chromatin inside the nucleus. Now you have to identify the correct combination of statements. Correct combination, if you ask, these are the combinations provided to you. A, C, D, A, B, D, B, C only, A, C only. So among these four options, which one according to you is a correct combination? Check out the statements here once again. What are the requirements of protein import inside the nucleus? I can see Sayanti Anusha has given the answer. Sayanti, you meant only statement A is correct? Okay, you are saying A, B, D. A, C, D, you are saying. Okay, A, C, D. Okay. Sharma, 2. Okay. A, B, D. A, C, D, 1. Okay. Okay, Rahul. Any other answers? Now, this is a very favorite part of CSIR. Every time you expect a question, like till the last exam also, if you go through the question paper, you'll find the question being asked from this NL, specifically the nuclear input. Very favorite of CSIR, and past also you will find the questions do come from here, and that too in the form of 4 marks. Okay, ABD. Okay. Okay, let's check out the answer. The correct answer over here is ABD. A small monomeric G protein RAN, a nuclear transport receptor that interacts with NLS on a cargo protein, and a guanine exchange factor bound to the chromatin inside the nucleus. So, this transport receptor is known as basically karyoferine receptors, we can say, or we can say alpha beta. Karyoferine receptors means basically involves import receptor and export receptors collectively. That receptor by structure we call alpha beta. Okay, let's have a look into the image. See, this is the image which gives you a clear cut idea about everything. RAN Jeff. Jeff means what? Guanine exchange factor. That means some kind of exchange takes place. What is that exchange? GDP is going out, GTP is coming in. So your RAN becomes RAN GTP. This is the active form. Where does it take place? Inside the nucleus. Where is Jeff present? Thither to the chromatin. Gap. Gap means what? GTP is activating protein. What does it do? It helps in the hydrolysis of GTP, giving rise to RAN GDP. So this becomes what? The inactive form of the RAN protein. That's why the correct answer is A, B, and D. Remember, RAN bound with GTP catalyzed by Jeff is the active form. RAN bound with GDP catalyzed by gap is the inactive. Okay, this is nucleus and this is cytosolic. Remember this points. Important from examination point of view. And this is how the import occurs. So this becomes your NPC, nuclear pore complex. These are the receptors, alpha and beta, which carries the NLS containing protein, which will be tethered to the cytoplasmic elements in the NPC, and the cytoplasmic elements will pull the cargo towards the nucleoplasm. This is nucleoplasm, this is cytoplasm. So the NLS containing protein has reached its destination, that is nucleus. Now what is the next thing? You have to translocate alpha and beta receptors back to the cytoplasm. So remember, beta receptors are taken care by RAN GTP, released inside or outside the nucleus, that is towards the cytoplasm. Exporting will carry the alpha receptors and releases into the cytoplasm. Okay, that is what the overall concept is. So as I told you, nucleus is a very important organelle. Be thorough with that organelle and the sequence of events that occurs during the protein transport. So see you again in the next session of Brahmastra series, where I'll be continuing with protein targeting, and if any pending questions from solute transport is there, that also will share it with you. Okay. Okay then, thank you so much everyone. See you again in the next session.